According to ancient Greek myths, one of the exploits of Hercules was the victory over the many-headed monster, the Lernaean Hydra. Fighting with him, Hercules chopped off one head after another, but in place of one cut down, 2 new heads grew. This fabulous myth served as the basis for the name of a new animal species discovered in the middle of the 18th century, Hydra. This animal was first discovered and described by Antony van Leeuwenhoek. This outstanding scientist, the inventor of the microscope and the discoverer of many microorganisms, found in the water of artificial channels on aquatic plants tiny creatures, animal icle, which either stretched or contracted, and at the same time moved their horns. Another similar creature was sitting on the belly of one animal Akula, attaching its tail to the first one. Leeuwenhoek's letter was printed in the Proceedings of the Royal Society, and thus the discovery of the Hydra officially took place in 1703. Few people then paid attention to this discovery, and mankind would not have known anything about the Hydra for a long time, if not for another discovery that literally excited scientists and the entire enlightened world of Europe in the 18th century. The Swiss Abraham Tremblay, the teacher of the children of Count Bentinck, conducted observations of wildlife with his pupils and discovered small green organisms in the ponds of the park, and not knowing anything about Leeuwenhoek's discovery, mistook them for plants. Believing that it was very easy to check this, he cut the object found by Tremblay and soon made sure that each of the fragments was restored to the whole organism. Tremblay's discovery gained great fame. His experiments were discussed in secular salons and at the French royal court. These experiments refuted the then prevailing belief that the absence of asexual reproduction and developed regeneration in animals is one of their most important differences from plants. Tremblay's original study was published in 1744. It was called Memoirs to the History of a Genus of Freshwater Polyps with Horn-Shaped Arms. Calling the organisms he discovered polyps, Abraham Tremblay also used the word hydra in relation to them, but in a metaphorical sense, since he found similarities between a budding polyp with 5 heads and the mythical Lernaean Hydra, with which Hercules fought. Carl Linnaeus liked this figurative comparison so much that in 1746 he proposed the scientific name for the genus, Hydra. Tremblay's experiments on the study of hydra regeneration laid the foundation for experimental zoology. So what is this amazing animal, Hydra? Hydras live in all kinds of freshwater habitats, including ponds, ditches, and slow-flowing rivers and streams. They are native to temperate and tropical regions. Hydra usually anchors itself to a substrate like a water plant by its foot. Hydra is one of the most ancient multicellular organisms. Its age is estimated at 600 million years. Freshwater Hydra is a single polyp, a very small animal, ranging in size from 1 to 1.5 centimeters, and the body usually does not exceed 5 to 7 millimeters in length, but the tentacles can stretch several centimeters. The body of the Hydra is cylindrical in shape, periodically opening at both ends. The upper opening, the mouth, is located in the center of the perioral cone, the hypostome. Around it is a corolla of tentacles. Tentacles most often 5 to 8, but sometimes more, up to 16. In some species of Hydra, the body is divided into a body and a stalk. The stalk, together with the tentacles, takes part in the movement of the Hydra. At the rear end of the body, the stalk, there is a sole. With its help the Hydra moves and attaches itself. Hydra has radial symmetry. The axis of symmetry connects 2 poles, oral, on which the mouth is located, and aboral, on which the sole is located. Several planes of symmetry can be drawn through the axis of symmetry, dividing the body into 2 mirror symmetrical halves. The body cavity of the Hydra, gastrovascular cavity, forms outgrowths that go inside the tentacles. Although it is usually believed that the Hydra has only one opening leading to the gastric cavity, the mouth, in fact, there is a narrow aboral pore on the sole of the Hydra. Through it, fluid can be released from the intestinal cavity, as well as a bubble of gas. In this case, the Hydra, together with the bubble, detaches from the substrate and emerges, holding upside down in the water column. In this way, it can settle in the reservoir. As for the mouth opening, it is actually absent in the non-feeding hydra. The cells of the ectoderm of the mouth cone close and form tight contacts, the same as in other parts of the body. Therefore, when feeding, the hydra has to break through the mouth again each time. Hydras feed on small animals, mainly crustaceans, Daphnia and Cyclops, but can also capture fish fry, therefore they are predators. Undigested residues are excreted through the mouth. The body of a hydra is a bag with a wall of 2 layers of cells, ectoderm and endoderm, between which there is a thin layer of intercellular substance, mesoglia. Mesoglia in hydra and other hydroid polyps in the form of a thin structureless plate, a basement membrane that lies between the ectoderm and endoderm. The endoderm lines the entire gastric, digestive, cavity up to the edges of the mouth. The endoderm contains several categories of cells. It is based on epithelial muscular digestive cells. In addition to them there are special glandular cells. These cells form the bulk of the body of the hydra. It has about 20,000 of them. They are cells that line the intestinal cavity and their main function is digestive. Glandular cells produce enzymes that break down proteins. The digestive juice of these cells enters the intestinal cavity, where digestion processes also take place. Hydra has about 5,000 glandular cells. In the ectoderm of the hydra, epithelial muscular, nerve, stinging and intermediate cells are located. The entire surface of the body, up to the edges of the mouth opening, is covered with ectoderm, consisting of several types of cells. Most of it is formed by cylindrical or cubic epithelial cells, the base of which, facing the mesoglia, extends upward and downward, along the longitudinal axis of the animal, into a long process lying parallel to the body surface. The cytoplasm of the process is differentiated in the form of the thinnest contractile fibers. These cells are called epithelial muscular. The totality of the processes of all such cells forms a layer of muscular formations at the base of the epithelium, coinciding with the longitudinal axis of the body. With their simultaneous contraction, the body of the polyp is greatly shortened. When the fibers of all skin muscle cells contract, the body of the hydra contracts. If the fibers are reduced only on one side of the body, then the hydra bends down in this direction. Thanks to the work of muscle fibers, the hydra can slowly move from place to place, alternately, stepping, either with the sole or with the tentacles. Between the bases of larger epithelial muscular cells are groups of small, rounded cells, called intermediate, or interstitial, eye cells. The hydra has about 15,000 of them. Intermediate cells are small, round, with large nuclei and a small amount of cytoplasm. These are indifferentiated cells. When the body of the hydra is damaged, they begin to grow intensively and divide. Intermediate cells can turn into stinging cells of the hydra body, except for epithelial muscular ones. Interstitial cells have all the properties of multipotent stem cells. It has been proven that each intermediate cell is potentially capable of producing both sex and somatic cells. Stem intermediate cells do not migrate, but their differentiating progeny cells are capable of rapid migration. Directly under the epithelium, stellate-shaped nerve cells are scattered, which communicate with each other with their processes and form a subepithelial nerve plexus. Thus, the nervous system of the hydra is at the lowest stage of development, has a scattered, diffuse character. However, even the hydra has thickening of the nerve plexus, around the mouth, on the sole and on the tentacles. According to new data, the hydra has a near-mouth nerve ring, similar to the nerve ring located on the edge of the umbrella in Hydromedusae. In total, the hydra has about 5,000 neurons. Part of the nerve processes approaches the skin muscle cells. The processes are able to perceive various stimuli, light, heat, mechanical influences, as a result of which excitation develops in the nerve cells, which is transmitted through them to all parts of the animal's body and causes an appropriate response. The presence of a nervous system allows the hydra to carry out simple reflexes. Hydra reacts to mechanical irritation, temperature, light, the presence of chemicals in the water and a number of other environmental factors. Hydra is the most primitive animal, in the nerve cells of which opsins, which are sensitive to light, have been found. Analysis of the Hydra opsin gene suggests that Hydra and human opsins have a common origin. A characteristic feature of the coelenterates is the presence of stinging cells in the integument. These cells serve as weapons of attack and defense. Stinging or nettle cells are located mainly in the ectoderm of the tentacles. Interestingly, they are formed from intermediate cells only in the region of the body of the Hydra, and then differentiated cnidocytes migrate to the tentacles. Stinging cells are the most numerous of all cell types. There are about 55,000 of them in Hydra. Such a cell has a stinging capsule filled with a toxic substance. A stinging thread is screwed inside the capsule. There is a sensitive hair on the surface of the cell. When it is irritated, the thread is thrown out and strikes the victim. After the filament is fired, the cells die, and new ones form from the intermediate cells. The Hydra has 4 types of stinging cells: penetrants, volvents, large and small glutinants. When hunting, volvents are the first to shoot. Their spiral stinging threads entangle the outgrowths of the victim's body and ensure its retention. Under the action of the victim's jerks and the vibration caused by them, penetrants with a higher irritation threshold are triggered. The spikes present at the base of their stinging filaments anchor in the body of the prey, and poison is injected into its body through the hollow stinging filament. A large number of stinging cells are located on the tentacles, where they form battery complexes. Cnidocytes are connected by desmosomes to the muscle fibers of the epithelial muscle cell. The large glutinants, their stinging filament has spikes but does not have, like the volvents, a hole at the top, seem to be mainly used for defense. Small glutinants are used only when moving the Hydra to firmly attach the tentacles to the substrate. The firing of Hydra penetrants has been studied using ultra-high-speed filming. It turned out that the entire firing process takes about 3 milliseconds. In the early phase of firing, Thorne's ejection, the speed of this process is from 9 to 18 meters per second. This allows the nematocyst to develop a pressure of about 7 gigapascals at the tips of the spikes, which is comparable to the pressure of a bullet on a target and allows it to penetrate the rather thick cuticle of the victims. Hydras do not have special respiratory and circulatory systems. The entire body surface participate in gas exchange. Gases and nutrients transported directly from cell to cell or by diffusion through the watery mesoglia. The Hydra also does not have excretory organs. Probably, the vacuoles that are in the cells of the Hydra play some role in the selection. The main function of vacuoles is osmoregulatory. They remove excess water, which constantly enters the cells of the Hydra by osmosis. Metabolic end products are excreted through the ectoderm. Hydra feeds on small invertebrates, Daphnia and other cladocerans, cyclops, as well as Oligochaetes cnidids, and fish fry. Prey is captured by tentacles with the help of stinging cells, the poison of which quickly paralyzes small victims. With coordinated movements of the tentacles, the prey is brought to the mouth, and then, with the help of contractions of the body, the Hydra puts on the victim. Digestion begins in the intestinal cavity. Abdominal digestion ends inside the digestive vacuoles of the epithelial muscular cells of the endoderm, intracellular digestion. Epithelial muscular cells, with the help of 2 to 5 flagella facing the intestinal cavity, mix food and adjust food particles, and with the help of pseudopods they capture them and draw them in. This is reminiscent of the nutrition of amoeba and other unicellular organisms. Digestive vacuoles form in the cells, where food is digested. Thus, in intestinal cavities, as in sponges, intracellular digestion of food takes place, a sign of primitive organization. However, along with this, the glandular cells of the endoderm secrete digestive juices directly into the gastric cavity, where digestion processes also take place, i.e. Hydra combines intracellular, characteristic of unicellular and lower multicellular animals, and intracavitary digestion, characteristic of multicellular animals. 2 ways of digestion in Hydra indicate the relationship of unicellular and multicellular animals. Hydra reproduces both asexually and sexually. During the summer, the common asexual method of reproduction by hydras is budding. Buds originate at the junction of the stalk and gastric regions. The bud begins as a hemispherical outpouching that eventually elongates, becomes cylindrical, and develops tentacles. The bud then pinches off and a new individual becomes independent. Freshwater hydra is a hermaphrodite, i.e. both male and female sex cells develop in the same individual. Some species, such as stalked hydras, are dioecious, and their fertilization is cross. In autumn, with the approach of cold weather, in the ectoderm of the hydra, germ cells are formed from intermediate cells, spermatozoa and eggs. The egg cells are located closer to the base of the hydra and look like an amoeba, and the spermatozoa are similar to flagellar protozoa and develop in tubercles located closer to the mouth opening. The spermatozoan has a long flagellum, with which it swims in the water and reaches the eggs, and then merges with them. Fertilization takes place inside the mother's body. The fertilized egg begins to divide, becomes covered with a dense double shell, sinks to the bottom and hibernates there. And in the spring, a new generation develops from the overwintered eggs. Hydras die in late autumn. In the so-called green hydras Chlorohydra viridissima, endosymbionts live in the vacuoles of endoderm cells. Green unicellular algae Chlorella vulgaris. This symbiosis gives a significant advantage to hydras when environmental conditions change, food is scarce. In the absence of prey, a green hydra may still be able to survive for about 3 months using nutrients produced through photosynthesis. This allows the hydra to tolerate starvation. Algae uses for its growth and development products of cellular metabolism harmful to hydra, carbon dioxide and ammonia. In the light, such hydras can go without food for more than 4 months. Feeding on Zoochlorella assimilates, while hydra artificially deprived of endosymbionts die without feeding after 2 months. Zoochlorella penetrate the eggs and are transmitted to the offspring of hydra transovarially. Other types of hydra in the laboratory can sometimes be infected with Zoochlorella, but stable symbiosis does not occur. It was with observations of green hydras that Abraham Tremblay began his research. At the end of the 19th century, a hypothesis was put forward about the theoretical immortality of the hydra, which they tried to scientifically prove or disprove throughout the 20th century. In 1997, the hypothesis was proved experimentally. The experiment lasted for about 4 years and showed no mortality among the 3 groups of hydras due to aging. It is believed that the immortality of hydras is directly related to their high regenerative capacity. Hydra is immortal not because it consists of immortal cells but because aged cells are constantly replaced by young substitutes generated from stem cells. Perhaps the most amazing feature of the hydra is the regeneration of its parts. If the hydra is cut in half, then each part is regenerated into a new hydra. Even if you pass it through a grater, dividing the hydra into 200 parts, then each part, under favorable conditions, can be restored into a whole animal. Even Tremblay's early experiments showed that the polarity of the fragment is preserved during regeneration. If the body of the hydra is cut across into several cylindrical fragments, then on each of them, closer to the former oral end, the hypostome and tentacles are regenerated, and closer to the former abral pole, the soul. In the early 2000s, an international team of researchers deciphered the genome of the freshwater hydra. It was found that the hydra has about 20,000 genes, almost the same number as in the human genome. At the same time, many genes of hydra and humans are very similar. In this regard, hydra is used to study regeneration and morphogenesis processes. Such studies are aimed at studying the mechanisms of stem cell differentiation, which can open up new perspectives in the fight against human cancer.